Hello, friends and colleagues. Could you please take your seats? We're going to begin the presentation in 5 minutes. Thank you very much. Sorry. This is just for the sound booth. How are we doing in terms of volume? Okay.
Yeah, I kind of want to get some.
Give it one more minute. Let's have a moment of understanding visually, and then we'll say, let's get started.
Okay.
Give it like people are still
Must be new. Yeah. Yep. Oh, no way. Amazing, Carl. Well, and I'm, a company dedicated to age-related and chronic diseases. Our first indication of my topics. Thank you. I'm very excited to. Yeah. No way. Wow. You have such a great foundation. Tanya's amazing.
All right. Thank you all for joining us today for the BioAge JP Morgan event. I am Eric Morgan, co-founder at BioAge, and we're here today to talk about muscle aging, which we're very excited about, in part because of some great phase 1b trial results that we recently had at BioAge. So we've gathered together a panel of experts to tell us all more about muscle aging, why it's really important for human health, and why we're excited about it, and what they're doing in this space that's really unique. So I'd like to start with a brief round of introductions, so we'll just go down the line so you know who is talking to you. And we'll start right here with Bill.
Hi, I'm Bill Evans, and I've been involved in muscle research and aging for almost 40 years. I was the first person to describe the condition sarcopenia many years ago. I postdoced at MIT in nutrition, was one of the initial lab directors at the Human Nutrition Research Center on Aging at Tufts University. Along the way, I was the fitness and nutrition consultant for the Boston Bruins and New England Patriots for about 10 years. I was also on the NASA Scientific Advisory Committee. I helped to design the Human Research Facility on Station. I was also director of the muscle metabolism research at GSK for about 5 years. I'm currently faculty at UC Berkeley and at Duke in the nutrition department at Berkeley and the geriatrics program at Duke University.
And Bill's also been a valued advisor to BioAge for a number of years now. So great, thanks, Bill. Yeah.
Good afternoon. My name is Anne Belian, or Anne Beline, whatever you prefer. I'll react to it. I established Rejuvenate Biomed in 2017 after 17 years working at Johnson & Johnson. I did jump, I followed my dream, and we are working in the healthy aging space, so we are focusing on keeping people healthy as long as possible And we do that by developing combination products, compounds, drugs basically, that we can administer to people that are suffering from age-related diseases. The first one being sarcopenia, so we are extremely happy to be here together with BioAge, and congratulations once again to all the work you already did.
Thanks, Anne.
Great. Hi, everyone. It's a really great pleasure to be here today on this panel, and thanks so much to BioAge for organizing this. I think it's a really exciting time. My name is Hannati Youssef. I am the CEO and co-founder of Juvena Therapeutics, which has been operating now for about 5 years. And I am a scientist by training with an accrued expertise in the biology of aging and mechanisms underlying tissue degeneration through about a decade of work, through my PhD work at UC Berkeley, followed by a 5-year postdoc at Stanford School of Medicine, where I was collectively really focused on measuring and understanding changes in protein signaling that occur systemically in blood circulation as well as locally in tissues, and how those changes with aging lead to what's known as a loss of tissue homeostasis and the onset of multiple degenerative symptoms, including the loss of our own body's stem cell function and progenitor function, and the ability to respond properly in the context of, for example, injury to regenerate and repair, as well as things like inflammation. And Juvena is based on many of those discoveries, and at Juvena we're dedicated to mapping secreted proteins to unmet medical needs through a computationally driven platform that enables us to mine proteins secreted by stem cells for their therapeutic potential and then translate them into engineered biologics. Excited to share more and engage in a really exciting discussion tonight.
Awesome, thanks.
And hey there, Carl Morris. I'm currently the CSO at Solid Biosciences, which is a company focused on gene therapy approaches for primarily Duchenne, but we just acquired another company and increased our pipeline. So my background, I'm a muscle physiologist by training, and I realized very early on, after getting an assistant professor position at Boston University in the muscle and aging research unit, that I wasn't cut out for academia. So I transitioned over to Wyeth and continued working on musculoskeletal, and Pfizer, where I did a lot of sort of muscle adaptation type work, looking at muscle loss, prevention of muscle loss, and then transitioned into the muscular dystrophies and neuromuscular disease, and I've been at Solid about 7 years now.
Excellent.
So a bit of an outlier on this panel here.
But Carl also has a strong interest in muscle aging, and we'll kind of be delving into the relationship along the way between muscle aging and muscular dystrophy. So wonderful. Thank you all for being here. So it's interesting. There's been a lot of activity and interest in muscle aging now, over past decades, but really in recent years, there's been a lot of excitement and a lot of momentum building in this field. And so I'd like to talk a little bit about that. But first things first, I'd like us to explore a little bit about what is muscle aging, and why is this such an impactful condition for people. And so Bill, if you could start us off and maybe tell us a little bit about that.
Sure, thanks. Well, I call muscle the longevity organ because it is related to almost every disease-related or age-related disease, including dementia and cognitive function. The loss of muscle occurs continuously past the age of 30 and accelerates in some people, but not in all people. And part of the issue related to sarcopenia is that regulators and others think that sarcopenia is a natural consequence of aging, and it's not. That's number one. It's really important to realize that maybe only 20% of the aging population should be considered sarcopenic. I think we're at the beginning of a revolution, because when I coined the term and described the term more than 30 years ago, I described sarcopenia as the age-related loss of muscle mass, which would predict distal outcomes like the risk of disability. And in that period of time, the field, in large cohort studies, have used a surrogate measure of muscle called lean body mass. And so there has been this implicit agreement that lean body mass is the same as muscle mass, and it's not. Recently, we have a new method that can now accurately and directly measure muscle mass. And I'll just, if you indulge me just for a second, it's called the D3 creatine dilution method. And it fundamentally makes the assumption that 98% of the creatine in your body is in muscle. So we use a deuterated creatine label. It's a capsule. You simply swallow it. It's mixed with all the creatine in your body, and then the beauty of the method is that creatine is converted into creatinine and lost in urine. So we can take a single urine sample and measure how much muscle you have. And one of my principal co-authors is right here looking at his phone, Dr. Steve Cummings. And we were able to initially put this method into the so-called Mr. Oss study, which is a large cohort study looking at osteoporotic risk factors for old men. And astonishingly, what we found is this method is strongly related to all aspects of function, including the typical geriatric functional assessment tools, while lean body mass has no relationship with these outcomes, including what's called appendicular lean mass, which is used as the surrogate measurement. Now we have longitudinal data from this trial that shows that muscle mass is independently associated with disability described by activities of daily living, IADLs, which are called instrumental activities of daily living, which is like balancing your checkbook. So muscle mass is related to a person's ability to balance their checkbook or to drive a car. It's independently associated with mortality. And I'll just say, when you control for physical activity, percent fat, exhaustion, functional status, self-reported health, weight change, weight, strength, and physical performance, it's independent of all of those factors. So muscle mass is a really powerful predictor of outcomes. So I think that we're at the beginning of a revolution in how we think about muscle, because until we control for the amount of muscle that someone has, it's difficult to say whether muscle quality changes with advancing age. And the method is now in a study that Steve Cummings, my friend, is the PI of, called the Study of Muscle Mobility and Aging. We've incorporated it into the Framingham Heart Study. We've incorporated it now into the Women's Healthcare Initiative, which is 10,000 older women above the age of 70, the Tobago Longitudinal Aging Study, and remarkably, a longitudinal study in boys with Duchenne dystrophy, which we think muscle mass may be a powerful predictor of function and disease progression. So we'll talk more.
Yeah. So Bill, clearly, I think the thesis here is that when you measure muscle accurately, it's a really strong predictor of all sorts of really important functional status and health outcomes in older people. The interesting thing about muscle aging is it's not very well known as sort of a clinical state or an indication, and yet at the same time, pretty much all of us know people who are deeply affected by muscle aging. It's something that many of our older relatives suffer from. So Bill, I wonder if you could just briefly give some examples of how muscle aging directly affects older people.
Well, of course. I mean, many of us, especially older people, and we'll talk more about this, go through episodic periods of inactivity. I mean, muscle is a very, very plastic tissue. It disappears at an astonishingly rapid rate in old people, and it's very hard to build back. So, for example, when older people gain weight, 100% of the weight gain is fat. But when they lose weight, 50% of the weight loss is muscle. So there are clear differences, and many older people that go to the hospital, with normal function, 10 days later, they can't go home because they're so debilitated. So and it's all centered on muscle. It's astonishing how rapidly the changes in muscle occur, and we'll talk a little bit more about the really fantastic data from BioAge looking at the effects of something as simple as bed rest, which, by the way, is the #1 therapy for old people in the world.
All right, thanks, Bill. So, I think all of us know, have a relative, the 80-year-old, who can't walk steadily anymore, who can't drive a car anymore, who can't live independently because of physical disability. This is clearly muscle aging. We also have relatives who've gone into hospital and come out much less functional than they come in. So this is muscle aging.
I have a great anecdote. So we were doing a study years ago looking at strength training, and one of our older subjects came in. He was pretty debilitated, and he told us this anecdote where his mailbox was about 200 yards away from his house. He went out to his mailbox and fell down, and there was nobody there at all, and he couldn't get up. And he spent almost 12 hours there not being able to get up. And we put him through a strength training program, and he was able to climb stairs quite easily. So again, muscle is very, very plastic and very responsive to what I think is a new generation of pro-muscle therapy. So it's really, we're in an extremely exciting time with regard to muscle.
Wonderful. Yeah, so that leads us to our next point. So muscle aging is clearly a major, major issue. And now we're at this time where there's a lot of very interesting, there's excitement about developing muscle aging therapies and a lot of interesting new approaches. So let's talk a little bit about that and how people are getting excited. And maybe, Anne, you could start us off with the European perspective on that. What's been going on?
Sure. And I completely agree. It's a lot has happened lately, and thanks to the work that has been done by pioneers like yourself, Bill, we are now at a stage where we're really ready to translate. So we're now really ready to translate the basic science into something that really can be a medication and that can be applied. That's one part, but the other part is definitely also the interest, the general interest. So for instance, in Europe there was IMI, which is Innovative Medicine Initiative, and they created a partnership, a public-private partnership between the European Union and the European Federation of Pharmaceutical Association. So it's a project that was called SPRINT, and the SPRINT project, what it did was actually, it started to look at people of the age above 70, and they looked at 1,500 subjects. And what they wanted to identify or understand was, now how are we able to measure if sarcopenia or physical frailty is present? So in that large study, they defined a couple of measurements that they would be looking at throughout multiple years, throughout 3 years to be exact. And they had one part of that group that they had put on a type of exercise, like 2 times a week. They had to go to a center, it was mild activity, and then 4 times a week they had to do some exercises at their home place. And then people were also being assisted in their nutrition and got personal guidance. The other group was just explained about what is healthy living and what can you do yourself to be healthy. By looking at these different measurements, they wanted to achieve 2 things. One was to define a better definition and clarify it a little bit in a way that also the physicians, but also the regulators could understand when does somebody have sarcopenia or does have physical frailty. The other thing was that they wanted to show that if you would measure these elements throughout that period, you could see changes. Perhaps not massive changes, but at least changes. Because if you can measure it, you can know it, and then you can also do something with it. So what I did is with this proposal, they also went to EMA. They asked EMA if they could agree with their approach, and EMA really bought into their methodology, but also to the way they wanted to analyze the data later on. This is, of course, a massive event if the regulators are really now talking with us and helping us to see what exactly would be needed for them to be able to approve a compound in that new area. The other interesting part in SPRINT is that it's very international. Yes, the study is conducted in Europe, but one of the elements, which is the SPPB, the short physical performance test, which is actually measuring if you have a good balance, if you can get up out of your chair very well, and how fast you can walk. This is actually something that has been defined also here in the US. It's coming from the group from Gralnik, and they also included that one. Because you see a very nice collaboration between Europe, US, and Asia on this topic. And you don't see it a lot in a lot of different age-related diseases, but this one definitely is there. And it is probably because indeed people are getting older, and we all know intrinsically that being disabled or not being able to get out of your house or to go to the store or to go to your friends, that really has a huge impact on any type of age-related disease that can develop. So I think really having the fact that we have both, also the National Institute of Health with the sarcopenia project, which was part of what Dr. Goralnik has published, having that community really trying to look at it all together from different perspectives and then coming to an agreement, this is really now the moment, I would say, for us to not just go, but run and fly and get this to the people that need it.
Absolutely, yeah. So, in the past, regulators have been a bit more silent on the question of muscle aging, and it's really just the increasing recognition of the real importance of this for everyone in the population as they get older. That's now led to a lot more explicit discussions by, EMA and FDA about this area. So that's definitely a part of what's going on here.
And you can see it very well also in the non-dilutive funding. So that's also a way on how you can see if there's new interest in an area, which is definitely there. So it should be able to help us all.
Great. And, another really exciting area Bill sort of already alluded to, which is this idea that as we increase the accuracy of our measures, if what we're trying to treat is muscle aging and we can't measure muscle properly, we're not going to do a very good job at treating muscle aging. And that's something that has been improving quite a lot in recent times. Bill sort of spent a little while talking about that already. And so, there's been advancement in, the way we measure muscle and the way we measure function. And there's also been advances in how we are targeting aging, in how we investigate biology and figure out what the best targets are to actually use to modulate this process. And we have, some companies here that are really doing interesting things in that, and in how to develop these therapies for muscle aging. So let's talk a little bit about that. So Hanadi, maybe you could start us off and tell a bit about how you guys are approaching this.
Sure, my pleasure, Eric. At Juvena, we're really taking this approach and discovery approach that's guided by the biology of stem cell signaling. And we are using the latest in AI technologies combined with computation, bioinformatics, and really multimodal data modalities, including various omics data sets such as proteomics, transcriptomics, and high-content imaging to really unravel the complexity of stem cell secretomes, looking at proteins secreted by stem cells and really this class of secreted signaling proteins, which, as we all know, are really actually the worker bees of our bodies, right? Proteins that are secreted by pluripotent stem cells or even other cell sources will circulate and are responsible for all sorts of biochemical reactions, from responding to pathogenic stimuli to binding and activating our own body's progenitor cells to promote better differentiation, tissue regeneration, and repair. And so when we set out about 4.5 years ago to build Juvena's protein drug discovery platform for predicting, screening, and really mapping and measuring the disease-modifying potential of proteins in human pluripotent stem cell secretomes, we chose to really build the foundation first focused on muscle aging and neuromuscular diseases, given that, like Bill really kind of drove home, muscle wasting with aging is really in many ways associated with all-cause mortality. There is such a relationship between the health of our muscle and other organ systems. For example, many folks end up passing away from different types of cancers, not because of the primary cancer, but by cachexia, right, and muscle wasting and atrophy. And really just this, shocking fact, right, that as you age, you have this accelerated loss of muscle mass, strength, and function. And unlike in these youthful environments where you're getting the right protein signaling cues, your ability to really recover from traumatic events or just even from just that day-to-day, environmental insult really goes down. And so at Juvenile, we actually were able to, really first focus on identifying key drivers of muscle-specific regeneration, identified, in fact, 13 different factors that can promote not only improved aged or dystrophic human myogenesis, but act to promote the survival and viability of new fibers and improve metabolism, and really mapping out these proteins that are driving muscle regeneration. We were then able to map them to multiple diseases within both the age-related muscle indications as well as dystrophies, that these proteins had the potential to reverse disease progression and really promote this tissue regeneration and in many ways rejuvenation. So at Juvenile, we're continuing to do that across different therapeutic areas, and a little later on in the panel, I'll talk a little bit more about how a drug that we're developing that's also really well funded by the California Institute of Regenerative Medicine, the NIH, going also to Anne's point on the excitement, that regulatory agencies, government agencies are seeing in the potential now of new biology that we're uncovering, us and many other companies, to now target some of the greatest unmet medical needs of our time, given the very few, if any, FDA-approved drugs for most muscle aging diseases and muscle wasting diseases.
Yeah, thanks so much, Nadi. Yeah.
It was a good point that I worked in a lot of diseases and also in sort of the atrophy space, and it's really about preserving muscle first. We would always sort of go after muscle, trying to preserve the muscle first, knowing that after that there are still complexities with the disease, that we're looking at that would have to sort of treat. But we wanted to stabilize the muscle and allow for sort of the longevity of the patients, so they would give them the opportunity to sort of get the next gen therapies that came out. So I think it's a really important point.
Awesome, thanks, Carl. So continuing with this theme of sort of new approaches, I was hoping, Anne, you could tell us about your approach to muscle aging.
Sure, with pleasure. Always happy to talk about it. So at Rejuvenate, we, like every company, has a little bit his own way of working. So the way we defined is that the data should tell us where to go. I'm also a scientist, next to my job that I'm doing as founder, CEO, all these other things that we all do, right? So we said for us it's really important. There are two important elements to us. It's a new field that we're heading into. So it means that we cannot have safety issues. That means that you need to work with compounds that are very, very safe to move forward. That doesn't mean that in the slipstream you cannot come with new stuff. We definitely will, but we will open up the front that way. So what we do is we take compounds which are out there, which we know already for quite a while, for which we have safety profiles in place, and for which we know that we can use it in a chronic setting and also in elderly. What we do then is we define or we investigate which are the different genes that they can up and down regulate, and we have a platform built that we can then map these compounds to. We selected what we call our list of important genes in longevity, and we map them to those. Then we map them to the pathways which are underlying, and then we choose two compounds that we can combine coming in from different angles, having a broad way of defining a mechanistic approach. When we see that this is possible, and of course when we see that we can also IP protect it, also not unimportant, I would say, then we move forward and we go into C. elegans. These are little worms. They're not little people, but we can learn a lot from them. There's a lot of conservation on the longevity pathways. So we look at how long they live, how well they move, how their head moves, how their tail moves, and so on. When we see that we have a positive impact there, we go to mice models. We always take two models. One is normal old mice. So we have a CRADA agreement working with the National Institute of Aging, where we look what's happening to mice which are on the compound for a very long time, two different strains, the two sexes, and so on. And then the other one is, the other model that we use is what we call an indication-specific one. So if we see, based on the information that we pull out of the in silico elements and that we pull out of C. elegans, we can define which organ might be the most interesting one. For the first compound, RGX01, it's sarcopenia. We're taking here also the stepwise approach. First, muscle disuse, acute sarcopenia, chronic sarcopenia. There are a lot of different ways to get there, and I'm sure my colleagues will be talking about those as well. So we really have to look at it from every different angle that we possibly can. Our phase 1 study is also ongoing. We will have the last subject last visit on the 30th of March. So we are excited for the April, May, June period where we will be looking at all the data, and so we're already planning to go off-site, lock ourselves up with the statisticians to look at them. So very curious what comes out. It's a little bit different than yours, but we are very complementary, I would say.
Absolutely. Yeah.
So our subjects are between 65 and 75, are in a cost for 2 weeks. We do measurements upfront, like a biopt, MRI, DEXA, name it, it's in there. Unfortunately, in that study, we could not add in the 3D3 creatinine because we were still in Europe. The next one, I shouldn't promise it yet, but we are definitely working on it. And so after they come out of the cast, we'll do the same thing, measurements on strength and the whole blood, muscle biopt stuff again. Another 4 weeks on the compound, and then we do everything again, and then another 3 weeks without compound on a personalized rehabilitation program. So we are so excited. It's really the year. It's really this year it's going to happen.
It's a great approach, yep.
Yeah.
So wonderful. So we'll touch a little bit more deeply on clinical approaches to targeting muscle aging in a little bit. But before we leave this particular theme, I wanted to also touch briefly on the sort of human cohort informed approach to muscle targets that BioAge uses. And Bill, as being quite familiar with the BioAge, I thought maybe you could comment on that.
Yeah, well, I mean, as Ann said, it's a fundamental question, and the question to ask, perhaps to start with, is why do we lose muscle as we grow older? I mean, it would be great if it were just physical activity. Of course, if you're less physically active, you lose muscle, but that's not the only thing. If it were, we'd just prescribe an exercise program and go home. And so there are multiple factors. There are neurologic factors. We know that we lose muscle fibers as we grow older, and there's a type 2 muscle fiber specific atrophy. But there are so many other factors that lead to muscle loss, and one of the approaches that BioAge has is that they have a cohort of people for which they have blood samples taken from early on, and then looking at changes over decades in physical function. So we can begin to look at perhaps what factors are associated with specific losses in function. And one of those, remarkably, was apelin, which is a myokine. It's a protein that is secreted by skeletal muscle during physical activity. And so what are the effects? Well, there was a beautiful paper published in Nature Aging on the effects of apelin that should be targeted specifically for old muscle. And some years ago, I was the principal investigator of a program project grant funded by the National Institutes of Health. And our question was, what happens when you put old people to bed for 10 days? And I had been involved in a number of studies funded by NASA. So when you take a healthy young person and put that person to bed for a month, they lose about 300 or 400 grams of muscle from their legs. When you take a healthy old person and put that person to bed, they lose a kilogram of muscle just from their legs in 10 days. It is an astonishing change. And it's all regulated by an almost 40% reduction in the rate of muscle protein synthesis. So there are these, and so why? Why are old people much, much more susceptible to these changes? One of them is that old people develop insulin resistance when they become immobilized to a far greater extent than young people do. And so there are tremendous numbers of factors. Muscle gets fattier when old people go to bed for 10 days. There's an increase in the amount of lipid, and that's driving changes in protein synthesis. And so 10 days of bed rest is equivalent to 15 years of aging for old people. And so you can imagine this sarcopenia that's moving in this direction, and then an old person becomes inactive for many reasons. Not just they go to a hospital, but they get depressed. They have some other health problems, and it goes down very rapidly. So this approach that BioAge has identified this really interesting molecule that may have profound anti-atrophy effects. And that's kind of what they did. And you may be familiar with the data, but it's really quite astonishing is that apelin agonist slows down or halts the muscle atrophy. It has a profound effect on the rates of muscle protein synthesis, which are driving those changes. So in many ways, we have a way to now target very specific aspects of aging that were collected in people decades ago. So it's a really exciting time. I think it's a nuanced, I think, targeted approach rather than what we were doing at GSK and Pfizer and every other drug company where we were using sledgehammers. I call them sledgehammers: anti-myostatin, anti-TGF-beta, biopharmaceuticals, selective androgen receptor modulators, and grelins that seem to affect mass, but very little effect on function.
All right, thanks, Bill. So another very exciting sort of advance is our increased understanding of muscular dystrophies and the relationship between dystrophies and aging, both on a biological level and also from perspective of how one can develop new therapies that target muscle. So I'd like to take a bit of time to explore that relationship between muscular dystrophies and what that can, the relationship between those and aging, muscle aging, and what that can teach us about muscle aging.
Yeah, well, there are multiple muscular dystrophies. Everyone sort of puts it in a bucket. Some of them are more atrophic, and some of them are very, very dystrophic, where the muscle actually gets damaged. There's a lot of fatty and fibrotic infiltration. But the key thing is that you generally are losing muscle mass over time, and it's being converted into something that is just not functional. In the case of Duchenne, this happens in boys, and by the time they're 13, majority could be sort of already becoming non-ambulant. So they lose the ability to walk. When you look at the MRI, you look at sort of an imaging or a cross-section of their muscles, it's completely covered. It's just fat. They're sort of tiny little muscle fibers that are left. And so we think about how best to sort of preserve that muscle, how best to sort of increase the strength. There are sort of obviously directed approaches. We're working on gene therapy to replace a protein that's missing. But beyond that, we have to sort of consider how can we sort of maintain that muscle mass. The other aspect of it is we have a patient population that we can sort of look at, we've followed, and sort of trying to identify how best to create a subgroup that is homogeneous. So when we actually test any of the drugs, that we actually sort of know what we should see in terms of natural history and whether we can sort of affect a change. And so I think applying that sort of logic where you can sort of select subgroups and identify, sort of do patient stratifications is sort of another thing that's sort of coming out of that rare disease space. There's opportunities to sort of look at very, very small patient numbers and sort of get the sort of results that you can sort of help lead you and guide you into the bigger studies and later stage studies. The other aspect, and I think we'll talk about it a little bit later on, is also patient-reported outcomes and how the specificity of the patient-reported outcomes, how the FDA is seeing that. I think you're best to talk about myotonic, but in Duchenne, we're using just a sort of a global sort of sledgehammer type patient-reported outcome. But now we're sort of working on defining a specific patient-reported outcome that will tell us and have sensitivity around what is important to the kids, what is important to the caregivers. And so that will allow us to sort of then go to the regulators and say, hey, look, these guys are happier, they're out playing with their brothers and sisters and their friends, they're not just sitting on the couch playing video games anymore. So we think we're doing something positive for quality of life. And I think those sort of lessons could be applied.
And I want to touch briefly on this aspect where, at least in some muscular dystrophies, they're really characterized by an increased susceptibility to damage during everyday activity, which occurs in all of us every day. We're always breaking and rebuilding muscle. And over the course of aging, people become less able to respond well to that damage. And that's something that is very, clearly accelerated in some muscular dystrophies.
Yeah, absolutely. And let the stem cell biologists talk about it.
Sure. No, yeah, I'd love to share more in that context
Yeah,
as
please
well.
go ahead.
So Juvena, we are developing a pipeline of biologics, but one of our biologics that's the furthest along in the preclinical development stage at the IND enabling and manufacturing stage is an engineered version of an embryonic protein that binds multiple receptors in order to really promote multiple muscle homeostatic mechanisms, including interacting and activating mTOR signaling with crosstalk with sphingosine 1-phosphate and BMP signaling to not only promote improved myogenesis in the age and dystrophic context, but also improve the maturation, viability, survivability of fibers, and improve metabolism and things like improved insulin sensitivity, which we know is also majorly deregulated with aging in the context of a lot of muscle-related diseases. And so we're developing it for myotonic dystrophy, which is actually the largest muscular dystrophy. It's an autosomal dominant muscle wasting disease that's characterized by a CTG repeat mutation in the DMP kinase gene, which is a core gene necessary for muscle homeostasis. We're just getting one copy of that CTG repeat mutation manifest in the disease. So it's really tough to target with things like gene therapies. Some approaches that are being developed are knocking down both the healthy and bad version of the disease. And it ends up forming, actually, when you express the DMP kinase gene with the CTG repeat mutation, an RNA hairpin loop that then causes sort of this stickiness that binds to splicing factors, causing aberrant splicing that then leads to downregulation and dysregulation of numerous regenerative pathways that are also deregulated in the context of aging. So when we were developing our JV161, based on our screening approach, we actually showed that it shows efficacy across age-related preclinical models and dystrophies. Right, JV161 was working in the context of aged, animals in the sarcopenic for sarcopenia, in glucocorticoid-induced atrophy, also for Duchenne's, and then we started testing other types of dystrophies, including myotonic. And what led us to actually myotonic dystrophy type 1, DM1 abbreviated, was really doing a thorough indication selection and prioritization analysis, looking at then the mechanisms of these several hits that we generated through this phenotype-based screening, and then realizing that the way that JV161 was working was in a manner that was not only targeting age-related muscle wasting, but some of these same mechanisms were also really crucial to the degeneration in certain dystrophies. And in fact, myotonic dystrophy is characterized by the FDA as a prodroid accelerated aging disease, where not only are they getting muscle wasting, but they're getting multi-system dysregulation, metabolic dysregulation, and it's a great way in our mind to get a biologic, right, approved through the regulatory process for a very focused patient population with really strong foundations, with really clear inclusion/exclusion criteria, and I think this could be a really great way and avenue for then us to expand and in many ways then further develop 161 and some of our other assets for these larger unmet medical needs where the clinical trials will be a little larger and longer duration.
If I can briefly add to that, because the two of you reminded me of something.
Sure.
Well, we're going to talk about indications, so.
No, I wanted to
Yeah, go ahead.
I just want to briefly touch on the patient-reported outcome, because we learned in the muscle field, we learned a lot from the more rare diseases and see how we could apply it in the other type of diseases. And one of the things is the patient-reported outcome. So in 2021, there was a paper published related to sarcopenia in this case, where it was described how a phase 2 and a phase 3 should look like. It's a recommendation, but it's a very interesting recommendation, and they took into account indeed things like what we mentioned before, the functionality, but also the muscle mass, like the D3 creatinine is also listed in there, and also the patient-reported outcomes. And they had two types that they included in there. There was a little preference for one. One is called the ARMLQ. Forgive me, I don't know exactly anymore what the abbreviation stands for, but it's muscle related. And it's a more generic one. And then they had another one, which is the SARCOL, which is a sarcopenia questionnaire, where they have limited it to 55 questions, and then they made another one with a shorter list, so where people really have to tell themselves what they feel and what they think. And these three elements will be important, because both FDA and EMA really are looking for patient-reported outcome. So this is really, thank you for reminding me.
No, absolutely. So, there are many ways to potential approval of new therapies, and one of the, FDA would love to know that, these therapies are really benefiting people subjectively, that they feel like they're healthier. And so this idea of developing PROs that we can use more specifically for muscle aging indications is, really interesting. And Bill, you were mentioning the FDA has some interest in that as well.
Yeah, perhaps the father of PROs, I don't know if anybody here knows Dave Sella at Northwestern University, but he developed multiple PROs in the cancer and function-related area. FDA has provided David a large grant specifically for sarcopenia. And he was asked to develop a PRO specific to sarcopenia, to conduct a clinical trial to look at the relationship between function and PROs in older people. So I think it signals to the field that FDA is interested in sarcopenia. EMA seems to be more interested in frailty. There is a crossover between these two, and I'm not going to bore you with the distinctions between the two of them, but they're both fundamentally the same syndrome. It's those older people that are weak, exhausted, have low strength. And so I think that we're at a new level, and one of the areas, again, this effect on muscle in a simple bed rest study, then ask the question, what is the indication? Where should we go if we have a drug that helps to prevent muscle loss in healthy old people? Well, we know that most of the people that end up in the ICU are old. We know that most of the people that died of COVID were old. And there was a really beautiful report in JAMA that just showed that old people that got and recovered from COVID were far more likely to be functionally impaired than younger people. So thinking about this, I think the BioAge approach is to think about the ICU, because in fact the rate of loss of muscle is nothing short of astonishing. Almost a kilogram a day in some older people. So if you can affect that almost in any way, you can have a profound effect on the health and recovery period of older people. The studies show that older people that go through ICU care almost never recover their lean body mass during that functional status. So if we can do anything to prevent the loss of muscle, and obviously these people are mechanically ventilated, their muscles are mechanically silent, and there's an enormous loss of diaphragm function, and so the diaphragm atrophies. And so if we can have an effect on any of those factors, I think that would be one of those endpoints that regulators would have a hard time saying, that doesn't mean anything.
Yeah, thanks, Bill. So we've sort of segued into this other very interesting topic, which is all of us are interested in muscle aging. There's a huge unmet need. And the long-term, really high-impact indication here is age-related sarcopenia or frailty, which everyone is vulnerable to as they age, and an increasing fraction of people suffer from, with each advancing year. However, along with being a massive indication and a massive unmet need, it's an indication where the trials are going to be very large and very long and very expensive. So strategic clinical development in this area is pursuing, a number of different tacts. And so we've actually, touched on a couple of these just now, just to kind of recap. So one is muscular dystrophies, right? This is a way to take an indication where there is a very severe need. People are severely affected. Declines happen over a short period of time, and therapies can show efficacy, over a very short period of time. And another one is these acute indications like, bed rest or, atrophy in the ICU, where, patients die in a short period of time if they can't be successfully extubated because of their diaphragm function. And so this brings us to this topic of what are the, how do we want to think about clinical development and, strategic approaches? And so, we've touched on a couple. I don't know, did anyone want to comment further on any of those topics, like the dystrophy? Is there anything else you guys would like to mention about using dystrophy as a, clinical development strategy?
Yeah, so I mentioned a little bit, but one of the good things about rare disease is you can skip a step. You just got to go from an early stage study and jump right into sort of your pivotal. And so it makes it a much more rapid development path. But you need to know what the outcomes are, and we actually are lucky that we have to sort of select a specific patient population. I think with aging, it's really identifying the sort of knowing what is translatable and what you can sort of, what specific sort of indication you can go after. I know sort of we looked at everything, and Bill, you're involved in it. We look at kidney, sort of like renal failure, like atrophy associated with renal failure, COPD. There are all these sort of different approaches, and they all have pluses and minuses. And I think now we're getting to the point where a lot more is understood, and particularly around the bed rest. There's translatability. We have casting models and unloading models and immobilization. There's sort of looking at the cancer cachexia. There's a lot of different approaches that could be taken to sort of become more specific in your patient population. And I think that's sort of coming from where, the rare disease sort of has that accelerated approach, and we really sort of think a little bit more. There's a lot more agility, and there's a willingness now, I think, from the regulators to be a bit more agile, at least in the first phase, and then they sort of help guide you, and I think that's coming along.
Yeah, if I could just say one thing. Having been at GSK for about 5 years, trying to develop thinking about sarcopenia was clear, and I won't go through the regulatory history because it's long and complicated, but the fact is that it was clear that no one was going to develop a sarcopenia drug in the early teens because of the regulatory hurdles. So most of the companies that were interested in muscle moved to cachexia, and that's muscle loss related to cancer and other indications. Now, what was interesting is that what's the largest cachexia disease? Well, the first thing you would think, I know someone with cancer, and I know what they look like. And so that was the pivot that many large pharma companies went to, is cancer. The problem is the FDA requires a functional improvement for whatever drug you may be thinking about. And fatigue and functional status in cancer patients is caused by a lot of other things besides muscle, chemotherapy being a principal one. And so some of those trials failed without careful consideration of what the real biology of the disease was. I think that we're now at a stage when we can think about aging specifically rather than cachectic diseases indications that are related to acute changes in muscle mass in a broad spectrum of older people. So I think, you know, if we don't get to a large sarcopenia indication in the next 5 years, that's okay, because I think what's going to happen is there's going to be, with the approaches that we've heard about, smaller indications that are going to be positive, leading to, you know, doctors starting to think differently about muscle and not just thinking about exercise and diet.
Yeah, thanks, Bill. Yeah, Nadia, go ahead. Yeah.
Yeah, sure. Just to add a few more points back on sort of the, the dystrophy side as well.
Please.
You know, I think what's really exciting, as was mentioned, was also the really accelerated translational process where, for example, we can now apply for orphan disease designation. We can get a combined phase 1-2 clinical study going, a smaller starting, patient population enrolled, and then with certain dystrophies such as myotonic, there is also a much better understanding based on recent natural history studies that were conducted in terms of different segments to consider for inclusion, exclusion, and patient segments, such as, for example, there is the geriatric kind of childhood onset form of the disease. There is also the adult onset, and it's a progressive disease where there is, multiple phases in this sort of triphasic process where at one point, year after year, there's an acceleration in terms of the loss of things like walking speed, grip strength, force vital capacity. And so clinicians, suggest that we could actually use some of those measurements in a point in the accelerated degeneration to potentially have the greatest potential effect on functional improvements that Juvenile, hopes to take advantage of in about 1.5 years when we're ready for our, with our IND for our clinical studies. And then importantly, just honing back in on the patient-reported outcome. That concept, what was I found fascinating as I started really learning and getting to understand myotonic dystrophy was there was a few studies so far with the biologic. It was called actually this clinical study, an IPLEX study. It was a combined, it was an endocrine therapy approach, very parallel and similar to kind of the type of paracrine endocrine signaling approach Juvenile wants to take. It was a IGF-1, IGF-BP3 complex that was being administered systemically. And what was, at the time they hadn't yet done the natural history studies. They didn't, everyone who just, had the mutation was, who would enroll and was able to kind of enroll in the study. So you had a lot of heterogeneity. And so what was really interesting and what many of the clinicians we spoke to who ran the study, said was that patients who were on the actual IPLEX drug were reporting personally these improvements in energy, in their metabolism and gastrointestinal, symptoms, in what they considered their strength, right? Being able to walk upstairs like they weren't able to before, being on the treatment. And everyone who was on the drug wanted to get that extension, the open label extension. But because of the heterogeneity of some of the functional outcomes, they weren't able to get that statistical significance, and it was like almost approved but not approved. But what the greatest thing that came out of that trial was then really this concept that what patients say really matters, and I think the FDA is really, and other regulatory agencies, appreciating the fact that with muscle symptoms that are so many times associated with so many other comorbidities, what patients actually say and feel really matters. And so now we have this patient-reported outcome metric that's going to be included in Juvenile's upcoming, for example, trial with our 161 fusion protein we're developing. And hopefully when we talk to the FDA later this year, we'll convince them that it's not just, of course we want to have those functional outcomes, and we're going to be very strategic with this, but that the cross-sectional area, the fiber typing towards type 2, the preventing the atrophy, the muscle, the actual lean, muscle mass. These are things, and even metabolism, biomarkers, various biomarkers are just as important. And we'll let you know how it goes, but I think, there is a lot of open-mindedness that we didn't see just a few years ago.
Yeah, go ahead, Carl.
And I just sort of reminded myself, a lot of activity monitoring is sort of taking place in the rare disease space, we were involved with a sort of a project where we didn't know what we wanted, but we just sort of stuck some sensors on the boys and had them walk around the hospital, eat lunch, and I was actually there for one of them, played a bit of rugby with one of the kids. And then put all that data into a bucket and just sort of figured out what Duchenne looked like. They identified specific markers in movement and activity. One was sort of how far they move their hands, right? And it's much, much less. And their gait was sort of a little bit off, and you could actually sort of detect it. So in a non-biased way, we are actually sort of looking for what does the disease look like. I'm not sure whether those things are being applied, but the approaches are out there now that can really sort of help that. There's actually now a company that's pushing for a specific activity monitoring marker called 95% stride velocity. It's so basically that you put a sensor on your ankle and you have the kids wear it for like a week, and they measure how, like, the range of speed that the kids move. And that is an incredibly sensitive measure. In the EMA, I think it's sort of approved as an endpoint, and they're really pushing hard here for the FDA. So those types of things, it could be definitely applied in aging, and because anything that is so you lose activity, the sensors and the technologies of now is just phenomenal.
Yeah, I can say that in our bed rest study, these are in healthy older people, 10 days after bed rest, or right after bed rest, we measured their physical activity. They were sedentary 2 hours a day more, just 10 days after so I mean, it's not a subtle change, it's an enormous change in a short period of time.
And importantly, it's objective.
Yeah.
And it's the same in our study. So we also have indeed the activity measurements in there. So we took 2 just to be on the safe side. So we have the traditional wear in here, but also the one on the leg. Because indeed, especially in sarcopenia, we know that the activity measurements, since we're still like early, but we know where we're going, we have now the capability of adding in more than you typically would do in a clean trajectory that is known, where you exactly know what you need to show. So we need to include just a little bit more, right, Eric, in all our studies than you normally do. So we have DEXA, MRI, ultrasound, we have them all, right? So, but it will give us so much information, and it will definitely help the field forward.
We have to
But
figure out
we
what to
learned-
do with all the data, but yes.
Yeah, we
Yeah.
learned a lot from
One of the other things I can say is that one of the things the FDA always asks for, if you're interested in, for example, mobility disability in older people, is how do you judge that? And one of the things that Steve's study is doing in SOMA, we often do that is asking the question, and how limited is your mobility. There's now like a short video where people can look at it of different like stick figures and say, oh yeah, that's me. And it seems to be strongly related to their mobility. It's just very, very simple, yeah, that's me, that's what I do.
So I think, what we're hearing here is one of the exciting areas of innovation is really what we're measuring in our clinical trials, and that's just critical for, advancing from, early stage to later stage trials, convincing yourself that you have a real effect here, and ultimately as registration endpoints, for regulatory bodies. And, we're making advances in sort of, accurately measuring muscle, as Bill was talking about, or measuring the synthetic rate of muscle. We're making advances in constructing PROs, asking the right questions of the patients to really determine if they're benefiting from therapies. And we're benefiting from technological innovation and activity monitors, where there's just a lot more variety of those available that are higher quality and perform a larger variety of measurements, than there used to be. So you can buy a relatively cheap device, and it measures, steps and high intensity activity and sleep. And so I think we're all very interested in these, and it's part of the movement that we're all taking advantage of to really kind of leverage into this new area of muscle aging. So we're reaching towards the end of our time here, and I wanted to, before we move on to the question period for the audience to ask you guys what your questions are, I wanted to, I guess first briefly, stop and say, were there any other sort of burning points anyone wanted to touch on before I get to my penultimate question?
Looking forward to the questions.
So my last question is for each of you. I'd like each of you to give a brief answer to what you think progress in this field of muscle aging could mean for human health in the next decade. You know, let's think boldly and see what might be possible. Yeah, we could start with Carl.
Well, I think Bill said it before, but you know, loss of muscle is critical for every sort of all our aging conditions. So I think that's what it'll give us. And that's a brief answer, right?
Awesome.
Sure. I mean, I think just, starting to see success in translation of, assets that are being developed by companies like BioAge and other companies that are really focused on, and like Juvena, hopefully, that are really focused on mechanisms of aging, really targeting the aging process itself to promote regeneration, to develop new types of therapies that could be applied to tissue-specific diseases, then even across, indications very systemically is going to be really exciting. And just seeing now some of these success stories, right, and promising data, I think is going to help hopefully usher more investment, more also non-dilutive funding, more amazing scientists and companies and collaborations that will, open doors for a variety of different modalities and therapies that rejuvenate tissues to rejuvenate lives coming to fruition and coming to market, and really excited to be part of that effort.
Great. Yeah.
Muscle aging is a very complex biological process which has a lot of hallmarks shared with the hallmarks of aging. So whenever we learn about muscle aging, we know that we will have to impact a lot of different things: mitophagy, autophagy, mitochondrial health, neuromuscular junctions, and so on and so on. So whatever we learn now in this area, we will keep our samples very nicely with us that we take from the clinical trials that we're currently doing in this aged population, so that when more knowledge comes forward, we can go back and see what exactly happened to all these different biomarkers that are currently being evaluated. So I'm very sure that everything we do in muscle aging will be helpful and contribute to the healthy aging community in general.
Thanks, Anne.
I think the prospects are going to be explosive. I call muscle loss an existential challenge for all of us. And I think it's going to take a lot, not just like people in this room, but until the public is aware that this is a diagnosis that something can be done about, will be very little advancement on the part of regulators. There has to be public pressure. I mean, for example, if a geriatrician measures the short physical performance battery in his or her office, they should be reimbursed for that. And right now, there's no reimbursement strategy at all for any measurement of function for older people. I think that's really critical. And the real question is, my hope is that in 10 years, you'll be able to give your mother a medicine that'll keep her out of a nursing home. That's what I think is going to happen. And I think once that happens, I think it'll be the biggest selling drug of all time.
Yeah, that would be transformative, Bill. Yeah, and I think that's kind of what we're all here for. So, all right, that's a great note to end. And I'd like to take any questions from the audience, and they can be general questions for the panelists, or they can be directed at a specific person. Yeah, go ahead.
Just a general question about sensors. So you guys mentioned patient-reporting outcomes starting to become important. Where's the FDA at on accepting sensors as an endpoint that can be used for approval?
So in our space in Duchenne, there's actually a file they've already submitted to it's the measurement. It's actually not the sensor. It's the measurement that they're using, which is the 95% stride velocity. And so it's already been submitted to potentially become a primary endpoint.
Great. And I think, yeah, next, yeah.
Thank you for this panel and for focusing so much on this topic. I am really curious about the field's application of properly designed
I can tell you, we published a paper in the New England Journal of Medicine about 35 years ago. Got incredible press. We took people between 90 and 100, and we put them on a high-intensity strength training program. They were at 80% of their maximal lifting capacity. In 10 weeks, we tripled their strength. It's amazing changes. And not just their strength, but their walking speed improved, their balance improved, their depression improved. So here we are 35 years later, and where is it? We know it works. There is no national program. I've lobbied for a national exercise program. There isn't one. There's not even reimbursed rehabilitation for an old person when they leave the hospital. So that's where we are. I think we know what works. It works and it doesn't work because nobody really does it. There's an infinite number of reasons why, and it would take another evening to address those questions of why, but it's not easy.
Can I also
I think
actually
what-
add just something to that, right? Like, no, I think a lot of elderly, frail, or sarcopenic persons don't always have the capacity to do exercise, right? Like, I mean, to make it a little, personal, like my late father had, unfortunately, multiple comorbidities, right? He had diabetes, his knees, eventually by the time he got into his 50s, 60s were so painful, it was really hard to walk. He had bad arthritis, but then because of his diabetes and cardiovascular disease, couldn't get knee replacement, and so often what you're seeing are that it's not always that kind of simple and straightforward for certain individuals, but if you can also find therapeutic interventions, biologics, small molecules, mechanisms in which you can preserve that muscle mass and strength, ultimately that greater eventual mobility will lead to things like improvements in cognitive function, improvements in cardiovascular function, in pulmonary function, and multiple comorbidities. So, that's something I think just to kind of keep in mind too, that it's important to have more treatments than just exercise and diet due to certain circumstances and comorbidities.
Well, and there's this aspect that you can take a physically active, exercising, sort of middle-aged person, and over the next 20 years, due to the course of muscle aging, as well as, insults, where they may be at bed rest for a period of time, and they become, much less able to do those activities that keep them active. And so, there's this barrier. So if we can prevent them from losing that muscle and function to begin with, that's the blue sky vision here. And if we can help them, restore it in acute episodes, that's sort of a near-term goal that would be,
That's
very
good.
valuable.
That's indeed a little bit my personal story. That's why I founded the company. So I was frustrated because I saw my father getting age-related diseases and he was not going in the right direction. So being at pharma, I thought, or I'm going to nag about it, or I'm going to do something about it. So I chose for the latter, perhaps not the easiest route. And what I indeed saw is exactly what you said. He had this comorbidity. So we put in as much physical therapy as we possibly could, as much as would be allowed. But you really see him deteriorating. And from the moment that he was not able anymore to really get out of his chair, which means that he was not walking anymore, we just see him melting away. It's painful, yes. Yeah.
Yeah, absolutely. And Joe was the next person I saw. Yeah. So the question was, since I'm not sure if everyone can hear, what if a muscle aging drug really worked? What would be the benefit to, to years of life or qualities that you would get from a muscle aging drug that really worked?
I mean, right now, it's interesting, NIH is designing what they call the ENHANCE trial to look at the combination of some amount of physical activity and a pro-anabolic therapy. So So it's hard to think of any one drug without some other ancillary effect. But the answer to to your question is people will be independent until they die. I think that's the goal. Well, it's going to be different between men and women, isn't it?
Yeah.
Which way? Men die a lot earlier than women do, so they have a much longer period of time in which they're physically challenged, I think.
Yeah, definitely want to emphasize that I think maybe some of the first drugs won't be magic bullets, right? I do think that there's going to be a lot of room for complementarity, for multiple types of treatment targeting, multiple different core mechanisms that really drive degeneration of tissues in our body with aging, but that we will see this really, first and foremost, this increase in health span, right? Folks like my father and Ann's father and others, and hopefully future generations, right? In the future, we'll have that ability to live those, not just your 40s, but your 50s, your 60s, your 70s, being able to travel and play and really live life to the fullest. And I think that will hopefully be one of the first things that we see coming out of some of the therapies that we're developing.
Great. Steve, yeah.
Imagine that you might have a drug that slows muscle mass loss in response to bed rest or inactivity. Try to imagine who would benefit. You'd like it to be the person with the lowest muscle mass, but would it be that it's more effective in people who have less activity, more sedentary, maybe less active, or even not effective than those who are active and maybe have less strength training? Who's going to benefit in all those who are doing the strength training?
So the question is, if you have a drug that prevents atrophy, muscle loss, yeah, prevents muscle loss, yeah. Is it going to be are the people who benefit going to be the ones who are sedentary or who are very active and training? Or both?
What was amazing when we did our bed rest studies, I assumed that the proportion of loss of muscle would be similar to what we saw in young people, but the magnitude would be smaller because they have less muscle to start with, when in fact, the loss is 3 times greater in older people. So the answer to your question, the people that would benefit the most are those people that are extremely sedentary and then develop pneumonia and have to go to the hospital or in the ICU. Those are the people for which the loss of muscle becomes catastrophic. For those people that are very physically active, I think they can sustain those losses probably multiple times and still be independent and functional. So I think it's the vast number of people who don't do anything.
It gets to a very interesting point, which is, what's your measure of success, for that question? And if it's just like loss of muscle and recovery from muscle, it could be that everyone benefits. What Bill's getting to is this point that once you pass a threshold, muscle affects everything in your life. And so, yeah, there is this threshold effect that's very interesting.
So screening for potential use or indication on this method, not necessarily.
The question is, how would you screen to figure out who's going to benefit the most from this? Yeah. And I think it depends on the therapy. I think you could screen for people who are at greatest risk and treat them. For us, we're going into the ICU, and everyone who goes into the ICU is at profound risk, especially the elderly. So they all qualify. So I don't want to take everyone's time too much. So we're kind of getting to the end of our time. We have time for a couple questions. And I see one there. And actually, I'm going to, after that, go to this region because I probably haven't been looking there. So go ahead.
I think the question really revolves around the anti-myostatin therapies, which have largely been ineffective and Carl can maybe also talk about that because he had an anti-myostatin therapy at Pfizer.
Yes.
Part of
And
the
it failed.
problem was that no one really understood the PK-PD relationship. There was never a PD marker of anti-myostatin therapy, so they never knew what the dose was, I think. And again, the idea that you can upregulate, so myostatin regulates amount of muscle, and it upregulates probably the synthesis of everything, and so it never really, even in smaller amounts or larger amounts, overall improvement in function. And I think that the new generation, I mean, there's been an astonishing amount of basic science in muscle biology in the last 10 years, and we have now far, far more targeted agents for very specific indications that you've heard here. I think the anti-myostatin, boy, there was a tremendous amount of interest there, but the animal data never translated to the human data.
Yeah, we tried hard, and I went into muscular dystrophy with the anti-myostatin, given that we couldn't sort of get, sort of a push into the age, any sort of muscle atrophy models. It's really about, like, everyone thinks about it as an endocrine, so it's circulating, where actually the muscle produces it, and then the muscle uses it. So it's probably an autocrine or a paracrine, so it's a very localized event, and we may not have been able to get enough of the drug at the site to really sort of drive change. And I still like it as a pathway, because as you said, it's very tissue specific, but I think we need to be a lot smarter.
All right, so our last question, as I said, all right, go ahead, yeah. Yeah, so the point was about, well, a couple points, but just recently about, strategizing to make sure that your clinical trial targets a compelling benefit for patients at a time when it matters to them. And I think that actually kind of dovetails with some of the points that have been made here. So with muscular dystrophy, people are experiencing, patients are experiencing decline over relatively short periods of time, and it's devastating for them. In scenarios like bed rest, again, they go into the hospital and they can lose function over a very short period of time, and that's really, again, devastating for them. If they've lost muscle over the course of 30 years, they may have forgotten what it's like to be able to do their prior activities. I think actually maybe it's something you wanted to talk about, Anne, because I think you're also interested in kind of approaching that at a lever moment when people are about to lose a lot of function.
Yes, exactly, and we completely agree with your comment. We need to measure things that we can measure objectively to be able to stratify. So we need to define which group do you want to get into your clinical trial. Before you can do that, you first need to identify which one it would be. Fortunately, there is a lot of work that has been done, like for instance the SPRINT trial, where it was clear that SPVB between 3 and 9 would benefit more than anything that is, between 3 and 6 would benefit more than anything above it. 3 to 9, well, the last part in that SPVB, which goes up to 12 points, right? that might not be the people that really will have the biggest benefit because they're procentually, because they're not doing that badly. The thing that's in the sarcopenia trials is the 400-meter walking test, which is indeed you can do it or you cannot do it, or you can do it faster or not as fast. Now for people, typically it's not important if they can do it really fast, so you also just want to know if they can do it at all. Because people don't, at some ages, might not mind that much if it takes them 20 minutes to get to that store or to get to their grandparents, but they need to get there somehow. So it's exactly these measurements that are being pulled in, and you need to establish upfront, indeed, do you put somebody in there that can do the 400-meter in 5 minutes, or 10 minutes, or 12 minutes? So that's exactly what we're looking into, yeah.
Just very quickly, I can say that for the first time, because we have muscle mass measurements in large cohorts in Framingham and Women's Health Initiative, we'll be able to do a GWAS specifically for muscle mass. Because you're right, there is a really large variability, even in the Mr. Ross study, which are just old white guys that are above the age of 80. It's incredible.
All right, well, thank you all for your questions. There's clearly a lot of interest here. The panelists are going to be hanging around. Our event is not ending. We'll be open for socializing and chatting and networking, I think until 8. So thank you so much all for attending, and I look forward to speaking to you in the crowd.